enzyme activity in phylos of some lichen species. Lichens are known for their resistance to extreme living conditions, largely due to their biochemical diversity, which has been actively studied in recent times, including their antioxidant systems. The study of antioxidant systems of lichens has increased, mainly due to the search for natural antioxidants for use in biotechnological processes. At the same time, studying the antioxidant activity of lichens as a complex indicator of organisms' response to changes in living conditions will help to understand the causes and mechanisms of lichen sensitivity or to changing environmental conditions. Our research objects included some of the most studied enzymes in the area, catalase and superoxide dismutase. The goal of our study was to investigate the activity of catalase and superoxide dismutase enzymes in the phylos of Atsana lichens inhabiting forest medium and subarctic communities at different ontogenesis stages. The object of the study was the epiphytic cyanobacterial lichen, Piltigera. Those photo beyond is a genus of nostoc cyanobacteria. This lichen most commonly inhabits moss at the base of aspen trees. It is on this species that we will focus our study of antioxidant systems. Samples of the phylos of the studied Piltigera were collected in the mid-taiga forest communities of the Kiwach Reserve and in the subarctic forest community of Biningis Reserve. But before moving on to the presentation of the main part of the research, it is necessary to talk about the phenomenon of reactive oxygen species, or known as ROS. ROS are partially reduced forms of oxygens with high reactivity. They have short lifespan and the property of interconversion. ROS are free radicals, and these radicals are highly reactive molecules or atoms with one or more unpaired electrons. ROS play an important role in the organisms of animals and plants. A certain optimal level of ROS is always present in cells as they are active participants in basic metabolism. The formation of ROS is genetically programmed, and under normal conditions, ROS are formed and transformed in reactions of basic metabolism, while under stress conditions, ROS accumulate and the plant's directions shift towards more intense secondary metabolism. The formed ROS can perform useful functions for the cell, such as participate in the regulation of cellular processes, have bactericidal action, and they can activate immune reactions. But however, this is only possible at low level of ROS in the cell. But at the high levels of ROS, oxidative stress develops, which leads to cell dysfunction. Biomolecules undergo free radical oxidation, leading to dysfunction in living organisms. Also, it might lead to cell death, development of various diseases such as cancer, and membrane lipids are susceptible to free radical oxidation, which leads to membrane damage. And of course, living organisms have specialized systems that control the level of reactive oxygen species. We will talk about them in more detail later. The antioxidant system is a defense system that includes systems that eliminate oxygen species or ROS and prevent their appearance, as well as detoxification systems that remove compounds damaged by oxidation with oxygen. Also, antioxidant systems are divided into two groups: enzymatic, which are proteins by nature, and oxidant by function, have high specified of action directed against a certain form of ROS, have cellular and organ localization specificity, and are used as metal catalysts, such as ferrum, cuprum, and others. And non-enzymatic, which neutralize ROS and make the plant a very plastic organism, are diverse in structure, nature, functions, and can functions in one direction or opposite directions. Cannot protect plants from superoxide radicals and hyperoxide. Those diagrams show an increasing trend in the number of studies focused on antioxidant systems of lichens. Here, one can note that the amount of research on enzymes such as superoxide dismutase, catalase, and peroxide, all of these antioxidants have a protein structures. Okay, let me show you. This slide shows the molecular structure of catalase and superoxide dismutase, as well as the electronic structures of some active oxygen forms. So let's move on our research. Specimens of Peltigera from 3 ontogenetic stages were examined. Virginal specimens are progenerative, phallus without reproductive structures. Generative specimens have apothecia, and senile specimens are aging phallus. Research was conducted in the laboratory of Aquaculture Research Center at our university to study the contents and activity of catalase and superoxide dismutase in lichen samples. This slide displays the average values of protein content and enzyme activity in phallus of this lichen on different ontogenetic stages. The histogram shows the activity of catalase, which breaks down hydrogen peroxide and superoxide dismutase, which neutralizes superoxide ions. These enzymes act in pairs, but as an evident, catalase activity is much higher. This is due to the fact that catalase has a known low affinity for its substrate, and it's only effective at high concentrations. As seen, catalase activity is high in virgin phallus and has low values in senile phallus. Similarly, in virgin phallus, catalase activity is higher in samples from North Tiger region as its protein content. It should be noted that the activity of catalase in Peltigera is very high, especially compared to higher plants and chlorobionc lichens. Peltigera prextata is a Tana lichen, while photobionc is a Tana bacterium that carries out the process of nitrogen fixation. The high activity of catalase in Peltigera may be related to the need to protect the nitral system from reactive oxygen species. As the conclusions to this research, it should be noted that the activity of catalase in superoxide dismutase is higher in virgin stages compared to generative and senile stages. The activity of antioxidant enzymes may be an indicator of metabolism activity at different stages of ontogenesis. No significant difference in the activity of catalase and superoxide dismutase enzymes were found among samples of phallus from different ontogenetic stages of Peltigera prextata Tana lichen in the mid and northern taiga forest communities. This may be due to the need for further research and increased sample size, or it may indicate the metabolic stability of lichens to the climatic gradient. And in the conclusion to report, it should be noted that given research for these natural antioxidants is becoming very relevant for the biotechnological processes. Thank you for your attention. Now I can answer your questions.
Yes, thank you for the great presentation. So questions to Nikita?